In this video, we're going to cover the structure and function of glial cells. We're going to break down the glial cells found in the central nervous system and the peripheral nervous system. Let's get started. Okay, if you've seen the neuron anatomy and function lecture, we covered how the nervous system is composed of two major cell types. We have the neurons and glial cells, or glia, or neuroglia. This is just a general term for many types of glial cells that we're going to break down in this lecture. The difference between the two is that glia don't have a direct role in information processing, but they do have several functions that are important for the healthy functioning of the nervous system. So looking at the structure of neurons and glial cells, neurons will have an axon and dendrites, which are used to transfer electrical signals, whereas glial cells don't. They're not directly involved in electrical signaling. They don't conduct nerve impulses. Instead, they help support the neurons and maintain the extracellular environment surrounding them. Glial cells are located within the central nervous system, which includes the brain and spinal cord, and the peripheral nervous system, which includes the nerves that connect the brain and spinal cord to the body's muscles and other tissues. In the central nervous system, there are four major types of glial cells. We have the astrocytes, microglia, ependymal cells, and oligodendrocytes. And in the peripheral nervous system, we have the Schwann cells and satellite cells. These are the cells we're going to break down in this lecture, and we're going to come back to this overview picture later on when we summarize everything, so you have a gorgeous diagram you can draw. So now let's subtract complexity and go through the stars of the central nervous system, the astrocytes. Astrocytes are highly branched. These astroboys are the most abundant glial cells. They are star-shaped cells. Astro means star and cyte means cell. So that's how you can easily remember their structure. They are the stars of the central nervous system, or the extracellular environment. Astrocytes maintain the extracellular environment. They are involved in the maintenance of blood-brain barrier homeostasis. They help regulate the content of the extracellular fluid by regulating ion balance. They also provide neurons with metabolic substrates for ATP production, and they also recycle or remove excess neurotransmitters around synapses. Let's break this down further. Let's start with the blood-brain barrier. During fetal brain development, astrocytes act as a framework to direct neurons to their appropriate final location, and also stimulate the formation of tight junctions between the cells that make up the walls of capillaries found in the central nervous system. And this establishes or forms the blood-brain barrier, which is a highly selective protective barrier between the blood and the brain. So the blood-brain barrier is the barrier or barricade between the cerebral capillary blood and interstitial fluid. It's made up of endothelial cells, these right here, the basal lamina or basement membrane, neuroglial membrane, and then we have the end feet or foot processes of astrocytes here. Now, between the endothelial cells, we have tight junctions, these blue regions, which only allow a small number of specific molecules to flow between the cells. There are also other cells that are found between the endothelial cells and the end feet here, such as pericytes. Alright? So, what does the blood-brain barrier do? Let's go through this. First, it prevents the passage of toxins and foreign substances from entering the central nervous system. It controls or regulates the flow of ions and substances between neural cells and the blood to maintain homeostasis. And this is where the tight junctions between these endothelial cells and specific transport proteins come in, because, like I mentioned before, only a small number of substances can pass through. These include oxygen and carbon dioxide, which are lipid-soluble molecules, and for larger molecules such as glucose and amino acids, they need to pass through transporters, these transporters here. So with that said, what do astrocytes do? What are their role? Astrocytes serve as crucial regulators in the development and maintenance of the blood-brain barrier, since they create a connection between endothelial blood flow and neurons to their foot processes right here. And so they help the blood's delivery of nutrients to the neurons, okay? They also release growth factors, which are important in the formation of tight junctions and also neuronal growth and maintenance. I also want to mention that astrocytes communicate with each other and with neurons through gap junctions, okay? That's the function of astrocytes in relation to the blood-brain barrier. Let's move on to the next function, which is the maintenance of potassium ions, or we can refer to this as potassium buffering. Extracellular potassium ion fluctuations happen in the central nervous system, and astrocytes are involved in the maintenance of extracellular potassium ion concentrations by absorbing or taking up excess potassium from the brain's extracellular fluid through ion channels and transporters. Let's go through this. We have a beautiful neuron here, this masterpiece, and neurons communicate with each other via action potentials, the flow of certain ions into and out of the neuron. We also have the diagram here. So during the resting membrane potential, there are usually more positively charged ions outside the cell than inside the cell. Potassium is usually more concentrated inside the cell, okay? And sodium is at higher concentrations outside the cell. And if you recall, during depolarization, we have voltage-gated sodium ion channels opening due to a stimulus, and sodium is going to flow inside the cell, changing the potential inside to more positive. Then the positive potential inside the cell during the repolarization, the falling phase of an action potential, this causes potassium ions to move out of the cell and into the extracellular environment here. So we have potassium ions in the extracellular space. And this is where the sodium-potassium pumps, or ATPase, these right here, come in. What they're going to do is they're going to return the potassium ions that leave back into the neuron, and they're going to pump sodium ions out. So these potassium ions here, they're going to pump 2 potassium ions back into the cell, and they're going to pump out 3 sodium ions. However, if the concentration of potassium in the extracellular environment is still significantly high, that the sodium-potassium pumps just can't handle it, it exceeds their ability to pump sodium and potassium in and out of the cell, this is where astrocytes come in to keep the ideal ion concentration around neurons. Why? So that we can maintain the normal neuronal excitability. Because what happens if you don't get rid of this excess potassium in this extracellular space here? There won't be a gradient anymore, which means less potassium will leave this neuron, and this can lead to hyperexcitability and eventually inactivation or decreased excitability. And we don't want that to happen. We want to maintain normal neuronal excitability. So this is where the astrocytes, the astro boys, come in, these purple boys, okay? Astrocytes have potassium ion channels, these right here. And what they do is they remove potassium ions from the extracellular fluid. Potassium can enter and exit through these channels. In this case, though, potassium ions are absorbed. They're taken up by the astrocytes and they are stored inside. And when we have an accumulation inside this astrocyte, it can also transport these potassium ions to other astrocytes via gap junctions. Because remember, they communicate with each other through gap junctions. Teamwork. Okay? And this maintains the extracellular potassium levels, which is absolutely incredible. Again, why is this happening? Because we don't want to interfere with the normal propagation of an action potential. All right? That's potassium buffering. Astrocytes take up excess potassium ions. Let's move on to the next function, which is energy uptake, storage, and delivery. I love this part. Glucose is the primary energy source for the brain. And astrocytes store energy in the form of glycogen, an energy reserve in brain tissue. So we have the blood vessels in red here, the astrocytes in purple, then we have the neurons. Glucose enters the brain via GLUT1 transporters, and they also enter astrocytes via the glucose transporter, GLUT1. And neurons take in glucose via the glucose transporter, GLUT3. Remember, there are 4 major GLUT transporters. We have GLUT1, GLUT2, GLUT3, and GLUT4. GLUT1 transports glucose to red blood cells and the blood-brain barrier. GLUT2, it's expressed in the liver. GLUT3, the brain, specifically the neurons. And GLUT4, we have skeletal and heart, muscle, and fat, okay? If you've seen the glycolysis lecture, there's a mnemonic that you can use to remember these GLUT transporters and where they are expressed. So we have glucose here, and like we mentioned before, astrocytes store energy in the form of glycogen. So we can break down glycogen to glucose to produce energy, and we can also store excess glucose. Now, glucose is converted into pyruvate by glycolysis. You already know the steps here. If you'd like a refresher, you can watch the glycolysis lecture. Love that lecture. And pyruvate enters the citric acid cycle or Krebs cycle, where it's oxidized to carbon dioxide and we produce NADH and ATP. Now remember that pyruvate also produces lactate. So this lactate can be transported to neurons and be used as an energy substrate. So it can be converted to pyruvate and enter the citric acid cycle to produce energy. And that's how energy is used and delivered. A simple overview. Let's move on to the next function, which is how astrocytes remove neurotransmitters. This is another gorgeous pathway and cycle. I love this one. Some astrocytes are closely associated with synapses, where they control the levels of neurotransmitters surrounding these synapses. They're able to detect the levels of neurotransmitters in synapses and can then respond by releasing molecules that influence the neuronal activity. They play a role in the maintenance and recycling of the neurotransmitters glutamate and GABA. Let's break this down. Here we have a glutamatergic synapse or neuron on the left side and a GABAergic synapse on the right, and also our astrocyte here in the middle. Let's slow down for a second because it can be quite overwhelming, but it's a walk in the park once you see the overview picture. Let's focus on the glutamatergic synapse first. The neurotransmitter that is released is glutamate. Glutamatergic equals glutamate. It's in the name. And so we have glutamate in this space. Glutamate is released, which acts on this postsynaptic neuron. What happens then is that glutamate can go back into the glutamatergic neuron where it's recycled and reused. But when we have an accumulation of glutamate in this space, glutamate is taken up by astrocytes. So here we have an astrocyte and it's going to enter the astrocyte where it's converted into glutamine by the astrocyte-specific enzyme glutamine synthetase, GS for short, using free ammonia. Now, here's the beautiful part. Glutamine is released into the extracellular space where it's taken up, it's going to be taken up by the glutamatergic neuron. It's going into this neuron where it's converted back to glutamate by glutaminase, Let's go through this again. So what are we doing? We're removing excess glutamate in the synapse and then convert it to glutamine. And then we can release glutamine back into the field where it's taken up again by the glutamatergic neuron and converted back to glutamate for reuse. We're recycling glutamate. How gorgeous is that, honestly? That's the glutamatergic neuron, glutamate. Let's take a look at the GABAergic neuron now. What happens in this synapse? What's the neurotransmitter that's being released? It's in the name, similar to the glutamatergic neuron. GABAergic equals GABA. We're releasing GABA. So these green molecules here are GABA. Similar to the glutamatergic neuron, GABA is released into the space and it's taken up into both presynaptic terminal, It's going back into the GABAergic neuron, and it can also be taken up by surrounding astrocytes when we have excess GABA in the field. In the astrocyte, GABA is converted to what? You already know, glutamate. We won't break down how that occurs. There are a few steps involved, but just know that GABA is converted into glutamate in the astrocyte. And then glutamate, this right here is converted to glutamine. And similar steps, we can release glutamine and transport it back into this GABAergic neuron where it can be converted to glutamate and then to GABA for this neuron to use. Alright, that's how astrocytes remove excess neurotransmitters around synapses. That's glutamate and GABA. Let's move on to the next glial cells, which are the satellite cells. These cells are the astrocytes of the peripheral nervous system. So similar to astrocytes, their function is to regulate the environment, the neural environment, to provide nutrient support and protection to these neurons found in the peripheral nervous system. They surround neurons in the sensory, sympathetic, and parasympathetic ganglia. They cover the surface of nerve cell bodies. They form these supportive capsules around nerve cell bodies located in ganglia. What is a ganglion? A ganglion is a cluster or collection of nerve cell bodies found outside the central nervous system. And so these satellite cells surround the cell bodies located in ganglia, the sensory and autonomic ganglia, where they provide, again, nutrient support and regulate the neuronal environment, similar functions to the astrocytes, Now, before we move on to the next glial cell, let's go back to our overview diagram and do a quick recap. So we have the astrocytes, the stars of the extracellular environment. Again, what do? They help form and maintain the blood-brain barrier. They take up potassium to regulate ion balance. They provide substrates for ATP production. They store energy in the form of glycogen, and they remove excess neurotransmitters, glutamate and GABA. So these are the main functions of astrocytes. Then we have the satellite cells, which support the cell bodies. These are the astrocytes of the peripheral nervous system. Let's now move on to the oligodendrocytes and Schwann cells, which are both responsible for myelination. Let's compare and contrast these two. They both cover neurons with myelin, which is an insulatory material. It's a protective layer composed of multiple layers of highly modified plasma membrane. The function of the myelin is not only to protect your nerve cells, but also to speed up their signal transmission and maintain the strength of the message as it travels down the axon. So it influences the speed of action potential conduction. So when the myelin sheath is damaged, the signal is slowed or stopped. Without myelin, the electrical impulses would be a lot slower. These signals would be delayed and disrupted. So these glial cells are crucial in providing support to neurons. In the central nervous system, the brain and spinal cord, oligodendrocytes are responsible for generating the myelin sheath. And in the peripheral nervous system, it's the Schwann cells. A mnemonic to help you remember this is COPS, C-O-P-S. Central nervous system, oligodendrocytes, PNS, Schwann cells. Let's compare them side by side. Oligodendrocytes are derived from neuroectoderm, whereas Schwann cells are derived from the neural crest. When it comes to the number of axons each cell wraps around, one oligodendrocyte branches and forms myelin around several axons. These ones can insulate up to 50 different axons at once, It can extend to 50 axons, whereas Schwann cells are capable of insulating only one axon, a single axon in the PNS, and along an axon, maybe as many as 500 different Schwann cells. The way I view this is that the oligodendrocytes are the players, whereas the Schwann cells are committed to one relationship or associated with one axon. Think Schwann, one. You are the one, And each Schwann cell in the PNS wraps around an axon, leaving small spaces between the myelin-insulated regions, This right here. And these are known as the nodes of Ranvier. So a small region of the axon membrane is in direct contact with the extracellular fluid. And the nodes are involved in the transmission of electrical signals along the axon. Now, lastly, what happens to these cells when they are demyelinated? We've already mentioned before that when the myelin sheath is destroyed, signal transmission is disrupted, and this can lead to physical or mental disability. In people with multiple sclerosis, MS, oligodendrocytes are damaged. This is a process known as demyelination. As MS is an autoimmune disease, the immune system attacks cells in the myelin sheath, disrupting nerve signals. So dysfunction in forming myelin in the central nervous system can lead to cognitive dysfunction. And when it comes to the PNS through Schwann cells, this can lead to Guillain-Barré syndrome, which is a peripheral demyelinating disease that affects the PNS. The immune system attacks neurons in the PNS. These are the cells responsible for myelination in the CNS and PNS, oligodendrocytes and Schwann cells. Let's go through microglia. Microglia are not actually neural tissue. They are the immune defense cells of the central nervous system, specialized immune cells. They are the brain's macrophages that, when activated, they remove damaged cells and foreign substances or invaders. They phagocytize bacteria and foreign invaders and remove anything that needs to be eliminated out of the central nervous system. Microglia are derived from mesoderm and have similar features to monocytes, which are a type of white blood cells that are frontline defense cells in various tissues throughout the body. So during fetal development, microglia move to the central nervous system and they stay there until an infection or injury activates them. Microglia maintain the neural environment. They monitor the central nervous system. They scan the central nervous system to make sure everything is good. They act as scavengers and eat materials that need to be eliminated. Let's go through this. We have a neuron here and a foreign invader that's causing damage. This is the site of injury. Now, microglia have a wide variety of receptors, allowing them to react to different stimuli, from neurotransmitters to cytokines to plasma proteins. And when microglia are activated, they move towards the affected area and respond to the injury and damage. They're going to release harmful chemicals to attack their target, as well as remove any cellular debris or foreign invaders by phagocytosis. They're going to eat them up. Now, activated microglia sometimes release reactive oxygen species that form free radicals. And the excessive release of these chemicals can lead to oxidative stress and may damage the neurons. So instead of protecting these neurons, it can cause damage. This can contribute to neurodegenerative diseases because nerve cells are being destroyed. Okay, one more thing, the only CNS cell type that may contract HIV, the AIDS-causing virus, is the microglia. And microglial dysfunction can lead to AIDS-related dementia. Okay, that's microglia. They act as scavengers, the brain's macrophages. They eat anything that needs to get out of the central nervous system. Let's move on to the last glial cell, the ependymal cells. Look at this masterpiece. Ependymal cells line the internal fluid-filled cavities of the CNS. They are simple columnar cells or simple cuboidal cells that line the ventricles, the walls of the ventricles. With this masterpiece, we have the brain interstitial fluid, neurons, and the oligodendrocyte, and the microglia here. I didn't put in the astrocytes. But going back to the ependymal cells, they form this membrane called the ependyma, which lines the spinal cord and ventricles of the brain. Let's go through this. Here we have the frontal view of the ventricles. I know I didn't draw the brain. I should have drawn it in. It would have made this a lot cooler, but oh well. This is the lateral ventricle and the other lateral ventricle. And this right here is the third ventricle and also the fourth. And the four ventricles of the brain are connected chambers that run along the spinal cord's central canal. Now, the ependymal cells line the ventricles, and they're involved in the production of cerebrospinal fluid, CSF, that fills the cavities of the brain, as well as the regulation of CSF, okay? The way to remember the function of the ependymal cells, think the CSF depends on ependymal. They secrete, circulate, and maintain homeostasis of the CSF to support neuronal function. They provide nutrients and provide protection to the brain and spinal cord. And CSF performs many functions, including allowing nutrients and other substances to reach the neurons, protecting the brain from injury, as well as removing waste products. Now, if we zoom in, ependymal cells have tiny hair-like structures on them called cilia and microvilli. These structures are found on the surfaces that face the open space, okay? And cilia contribute to the flow of CSF through the ventricles. How do they do this? These hair-like projections move or beat in a coordinated way to promote the directed flow of CSF. This helps circulate the cerebrospinal fluid throughout the cavities of the brain and spinal cord, bringing nutrients and other substances to neurons, and as we mentioned before, filtering out waste substances. Okay, so going back to our overview diagram, ependymal cells create barriers and they secrete and regulate CSF. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating!